Good evening, everyone. We'll wait for a few more minutes to let others join and then we can start today's session. I guess we can start. Let me just share my screen. Alright, I hope you can see my screen and you're able to hear me. Okay, so welcome back. Welcome to the 11th session of NPTEL online course Drug Delivery Principles and Engineering. Okay, thanks for letting me know. Right, so we can start with some of the points that were discussed in this week's lectures. So we started this lectures with gene therapy. So what exactly is gene therapy? So first of all, we all of us know, right, what genes do inside our body. They code for proteins, and in case some of these genes are defective or some of these genes are missing in someone's body, then that could create problems, because then those proteins would not be made, which could be part of which could be enzymes, which could be important parts of the building blocks of the body. So in this case, what we can do, one alternative for us is to keep providing proteins for the rest of the life of this person, but that becomes very cumbersome and is not a very practical solution. So to treat such genetic defects, we make use of gene therapy. So what we can do is we can take the gene which codes for the protein. In case the person is missing this gene, we can use this gene, use a gene that actually codes for the protein, put it into the patient's body, and then let it integrate into the cells, and then these will then keep on producing the protein. So that's one alternative. Or we can try using some gene therapy principles to rectify a gene that has gone wrong. So maybe this gene is producing a very different protein, it's producing the wrong protein. So in this case, then we can rectify the gene using gene therapy principles. So these are ways in which gene therapy helps in rectifying a disease or a genetic disorder. Now there are various different barriers to gene delivery. You cannot just take a gene, insert it, let's say inject it into a patient's body and hope that it will produce the protein. So what are these various different barriers to gene delivery? These could be, first of all, you've injected the gene, then it'll come in the serum. There are a lot of enzymes in the serum which will try to cleave this gene. So once so this gene is nothing but a sequence of nucleotides, right? So once it gets chopped up, you no longer have a gene which could reach to the cells. So that's one barrier. Then let's say it somehow escapes from all of these enzymes and reaches the cell. Now it has to actually enter the cell. So now we all of us know that the cell envelope, we have a cell envelope, right? And it is not very permeable to charged molecules, and your gene has a negative charge, prominently a negative charge. So your gene has a barrier entering into the cell. That's one thing. Then let's say it enters into the cell, then you have a lot of endolysosomal enzymes. So we know that in the lysosome we have a very low pH. So in this case, the gene can, there are chances that the gene will get degraded because of these enzymes. Right, and let's say all of this, the gene that you have delivered is able to escape from all of these enzymes, from all of this serum enzymes, endolysosomal enzymes. It is permeable into the cell. Now it has to reach the nucleus, because that's where all of your transcription happens. Your gene gets converted to mRNA. So it has to be transcribed for it to be made into a protein. So now the gene, it's already into the cytoplasm. It has to enter into the nucleus, and so it means it has to pass the nuclear membrane. So there are a bunch of different barriers to gene delivery. So that's why we cannot just deliver a gene like that. So to deliver gene, we have two different carriers. First is viral based vectors. So we know that viruses, what do viruses do? They inject their genetic material into a cell, and they could integrate it into the cell's genome, or it can exist as an extra chromosomal DNA, and in that way they just use the cell's machinery to make more proteins. So we can make use of these viral vectors. We can engineer viruses to infect the cell that we want and deliver the gene that we want the proteins that we want the cell to produce. So what are the advantages of having viral based vectors? First of all, it has a very high transfection efficiency, because these viruses in nature, they are already designed to deliver genes. So they have a very high transfection efficiency. They're very cell specific. So we can do targeted gene delivery using virus vectors. And also they have evolved mechanisms of endosomal escape. So whenever a virus infects a cell, most likely that their genome, the genetic material that the virus has, will also enter into the endosome, but they are naturally engineered to escape from this endosome. So we can make use of that. So these are advantages of using viral based vectors for gene delivery. Now we also have certain disadvantages with using viral based vectors, namely strong immune response, because now this is a virus, it is infecting a cell, injecting the DNA material. There is a chance that the cells might, the body might have a very strong immune response against this vector. It might activate oncogenes, because we now don't have a control of where that gene is going to be integrated into the cell. It can integrate anywhere, and in that process it can activate oncogenes. Oncogenes are nothing but genes that lead to cancer. Also, we have a limitation on the size of gene that we can pack into the virus. The virus has a very limited size, so we have limitations of how big of a DNA you can put into the, how big of a gene you can put into the virus. And also chance of viral disease, because although we can engineer to minimize this, there is always some amount of chance, some possibility that the viral vector that we are using could cause disease in the patient's body. So that's about viral based vectors. We have something known as polymer and lipid based carriers. So what we do is we encapsulate our gene into a polymer and then deliver that. Advantages: it has lower risk of immune response, because now it's not a biological living material that can infect the cells. This is just a polymer which is encapsulating our gene. There is no size limitation, because we can now engineer particles to hold how much ever amount of gene we want. No integration in genome, so these particles will just deliver the gene into the cell or into the nuclear membrane. They will not integrate this gene into the genome, so there is no chance of activating oncogenes. But disadvantages with these carriers: it has low transfection efficiency. It is definitely not as high as viral based vectors, because we have synthetically made these particles and we just rely on how they are going to deliver the gene. So definitely low transfection efficiency. They could be toxic at higher doses. So if we want a very high transfection efficiency, we might think that we can deliver a lot of particles, but they start becoming toxic at higher doses because of the polymers that we use. And also they'll have poor endosomal escape, right? They don't have any natural tropism to escape the endosome. So these are certain disadvantages of using polymer or lipid based carriers for gene delivery. So that's what we saw. We also have questions regarding this, so that will make the concepts more clear. So, what kind of polymers can we use? So, the most prominently used polymers for doing gene delivery is cationic polymers, which have a positive charge. Now, why cationic polymers? Because our genes are negatively charged. These are DNA molecules. They'll be negatively charged. So, if you use a positively charged polymer, more chance of encapsulating the gene or adhering the gene over the polymer particles because of the positive negative interaction. So that's why you use cationic polymers. Some examples of these are polyethylene imine, PEI, chitosan, dendrimers, etc. We'll also see why these polymers are useful in delivering gene, because they have proton sponge effect, which helps them escape the endosome. We'll look at this concept in one of the questions as well. Next concept that was discussed was using DNA as vaccines. So this becomes advantageous because it triggers both humoral as well as cellular immunity. Humoral meaning it triggers the B cell immunity, the immunity that leads to formation of antibodies against the antigen, and also your cellular immunity, that is nothing but your T cells directly showing their immune effect, directly killing the because they can identify the antigens. So that is one concept of using DNA as vaccine that was discussed in this week's lecture. Next, you have cancer vaccines. So these are vaccines against cancer. They could be prophylactic or therapeutic vaccines. What does prophylactic mean? This is like giving vaccine before the cancer has occurred. So this is prominent in some cases wherein some of these cancers are caused by viruses. So in that case, it is possible to make a prophylactic vaccine for cancer caused by a virus. And also there are therapeutic vaccines. So what are therapeutic vaccines? So for cancer, so these are nothing but you introduce certain antigens to the body which you think are going to, so you have your cells, right? Why is cancer caused? First of all, these are your own cells which have started growing at a very abnormal rate, and that's why they turn cancerous. And your body's surveillance system, the immune system, it is very efficiently designed to get rid of these cancerous cells. But sometimes what happens is some of these cancer cells escape from the immune surveillance, and in that process they start growing at an abnormal rate and they cause cancer. So in that case, we can use certain therapeutic vaccines. These are nothing but to strengthen our immunity against such cancerous cells. So these could be your autologous tumor cell vaccines, meaning you take the cells from the patient's body, the antigens are taken from the patient's body and then delivered as a vaccine to the same patient. Or it could be allogenic tumor cell vaccine, meaning you take these antigens from an allogenic donor or a different person, or cell lines which are grown artificially in the labs, and that's where you get your antigens from, and then you use that as a vaccine. So both of these, autologous and allogenic, have their own advantages as well as disadvantages. So these are some of the concepts discussed in this week's lecture. So we can then move on towards some of the assignment questions, if everybody is okay with that. In case of doubts, I can stop and we can go over some of these concepts again. If not, then I'll just move on towards the questions. I hope you can see the question displayed on the screen. So the first question is, what is gene therapy? We have to choose one correct answer from these 5 options. If anybody knows the answer, please let me know. Nonetheless, I'll wait for a few minutes for you people to read as well as go through the options, and then we can discuss. If anybody wants to answer, you can just unmute yourselves and let me know, or you can also write it in the chat box if you're comfortable with that. Okay, somebody is answering option 2. Thank you for responding. We'll discuss in a while. Okay, let's look at the answer. Yeah, whoever answered, you're perfectly correct. The correct option is option 2, treatment of diseases caused by genetic defects. So we were just talking about this, right? So gene therapy means you can treat a disease by introducing some genetic material into our cells. So these genes could be, let's say there's a missing gene or a defective gene. In that case, you can introduce a gene that actually codes for the correct protein. That is one way to do gene therapy, or we can use gene therapy principles to repair a malfunctional gene. So the gene is already there, but it's not coding for the right protein. So in that case, you can use gene therapy to correct that particular gene, or it could produce a therapeutic protein that helps to treat the disease, or we can use gene therapy to vaccinate the patient against an infection. So these are number of ways in which we can do gene therapy. Considering the options here, what is gene therapy? It is treatment of diseases caused by genetic defect. So that should be pretty straightforward. If you people have any doubts here, we can stop and go over this again. If not, then I'll move on towards the next question. Okay, let's look at the next question then. Next is, which of the following cell types are not considered professional antigen presenting cells, and we have to choose two correct options. So take your time in reading through the options, and if you people know the answer, please let me know. Okay, somebody's answering 2 and 5. Thank you for responding. I'll wait for a few more minutes. Okay, someone else has also answered 1 and 4. Thank you for responding. I suggest you please read the questions carefully because we have to select cell types that are not considered antigen presenting cells. Alright, let's look at the answer. So, whoever answered first was correct. The correct answer is neutrophils and T cells. So, as I said, please read the questions carefully. We have to select which of these cells are not antigen presenting cells. So, we have talked about this in the previous lectures as well, right? So, your dendritic cells and macrophages are actually the professional antigen presenting cells. They are present in the tissue and they do a surveillance around these tissues to see if they see anything foreign or any changed, any mutated cell. So in that case, these two are the antigen presenting cells. Your T cells are they could be helper T cells, cytotoxic or regulatory T cells. They are involved in directly removing the mutant cell, and your B cells are responsible for your antigen presentation as well as antibody generation. So the question is, which of these are not considered professional antigen presenting cells? So the correct answer is your neutrophils and T cells. T cells are not antigen presenting, neither are your neutrophils. Is that clear? Okay, I'll move on towards the next question then. Next is, which of the following are not advantages of non-viral vectors? We have to pick two correct options. We discussed this in the introductory slide as well, so if you people know the answer, do let me know. If not, we'll discuss. Again, we have to select what are not advantages of non-viral vectors. Okay, someone has answered 5 and 6. Thank you for answering. Anybody else? Okay, let's look at the correct answer. The correct answer is high transfection efficiency in natural tropism. So, whoever answered absolutely correct. These two are actually disadvantages of non-viral vectors. So we talked about this, right? So to use genes to deliver genes, we can use two different carriers. It could either be viruses or it could be polymers and lipids. These are non-viral vectors. So each of these have their own pros and cons. So the viral vectors, the advantages are they have high transfection efficiency, they have natural tropism, that is the ability to infect different cells, and they have evolved mechanisms for endosomal escape. They also have natural transportation mechanisms of DNA directly into nucleus. The disadvantages of viral vectors, they can generate strong immune reactions against viral proteins because we are going to use the entire virus, so the body can generate reaction against the viral proteins. There is a possibility of chromosomal insertion and oncogene activation. It's a complicated synthesis process because you have to engineer the virus vector in such a way that it delivers your gene, and we have a limitation on the gene size. So if you just look at the question again, which of the following are not advantages of non-viral vectors? The correct answer is high transfection efficiency. It's not an advantage. And natural tropism. Is that correct? I hope there are no doubts here. Okay, let's move on towards the next question then. Yeah, so the next question is, which one of the following is not true for chitosan as a gene carrier? So chitosan is one of the polymers which can be used to deliver genes. So we are supposed to answer what is not true for chitosan as a gene carrier. Okay, somebody's answering option 3. I guess you're correct. So yeah, so let's talk about chitosan. So as I said, it can be used as a gene carrier. It's a cationic biopolymer which can complex with the anionic DNA. So that's why we use cationic polymers, right? Because they're positively charged. Your DNA is negatively charged, so they can complex together. It's a natural polysaccharide, so it is essentially non-toxic in nature. Compared to synthetic polymers that we could use as gene carriers, chitosan is relatively non-toxic because it's a natural polysaccharide. It does not create any immune reaction. As a result, it's biocompatible and it's very suitable for mucosal delivery because it's mucoadhesive. So these are the advantages of chitosan as a gene carrier. So if you just go through the options, we are supposed to list what is not true for chitosan as a gene carrier. Is it a cationic biopolymer? Yes, it can complex with DNA. It is non-toxic, non-immunogenic, it is mucoadhesive, and it's a natural polysaccharide. So what is not true for chitosan as a gene carrier? That it is toxic. So that's why option 3 is correct for this question. Is this clear? Okay, let me just go back to the previous question. I think we did some mistake here. Okay, the question is, which of the following are not advantages of non-viral vectors? Okay, yeah, so we were talking about non-viral vectors, so I accidentally by mistake I explained you the viral vectors. But if you just talk about non-viral vectors, so we are supposed to list disadvantages of non-viral, which of these are not advantages of non-viral vectors. So that's why the correct answer was high transfection efficiency in natural tropism, because these are advantages of the viral vectors. So your non-viral vectors will not have high transfection efficiency since they are synthetic in nature. They also don't have natural tropism. We cannot infect multiple cells with this. So that's why this is the correct answer. I hope I have not confused you people with this question. If it's confusing, I can visit this back at the end of the assignment. If anybody has doubts here, we can come back to this question later on. Do let me know about this. Okay, so we are clear about chitosan. Let's move on towards the next question then. Next is, what kind of material is preferred for gene delivery? I think we were just talking about this, but nonetheless, I'll just wait for a few seconds here and please let me know if you have the answer. Okay, let's look at the answer. This was pretty straightforward. We were just talking about this. Cationic polymers are preferred for gene delivery. Why? Because DNA itself is highly negatively charged and it can form complex with DNA. One can modify them to attach targeting ligands. It can be endocytosed by the cells because that's what you need for transfection. And the most commonly used polymers for this is PEI, polyethylene imine, chitosan, and dendrimers. So to answer this question, what kind of material is preferred for gene delivery? It's cationic polymers. Clear? Okay, let's look at the next question. Next is, what are not the objectives of gene therapy? Again, we have to choose two correct options. So please read the question carefully. We are supposed to select what are not the objectives of gene therapy. Anybody would like to answer this? Have you already? Okay. Somebody's answering 4 and 6. Vaccinate a person against an infection, cause dysfunction at a cellular level. Okay. Anybody else? Thank you for answering. Okay, let's look at the answer. So the correct answer is last two options. So one of your options was correct. Whoever is trying to answer. So what are not the objectives of gene therapy? We already know what are the objectives of gene therapy. So we can use gene therapy to replace a missing or defective gene. We can use it to repair a non-functional gene. We can use it to produce therapeutic proteins that are going to help treat a disease, or we can vaccinate a person against an infection. So all of these are the objectives of gene therapy. The only two options that are not objectives of gene therapy are to cleave healthy genes already present to generate haploid cells. No, we don't want to do that. Cause dysfunction at a cellular level. Definitely don't want to do that. So these two are not the objectives of gene therapy. I hope this is clear. We have been talking about gene therapy a lot. It is also there in this slide. You can go over the presentation later on as well. I'll be sharing it with you people. So as far as this question is considered, I think it's pretty straightforward. And so I'll move on towards the next question then. Next is, what property of the polymer causes proton sponge effect? So we have discussed about proton sponge effect in some of our initial sessions as well. If you people remember it or you know the answer, please let me know. If not, we'll discuss. Okay, anybody? All right, let's. Oh, somebody responded. All right, let's look at the answer. So the correct answer is presence of secondary and tertiary amines. So we have also seen this in case of some polymers that we can use to deliver drugs and help them to escape the endosomes. So the same concept applies here. So what happens is, if you use polymers which have secondary and tertiary amines, what happens is these particles are uptaken through endocytosis, right? So the cells are going to uptake these particles and they're going to end up in lysosomes or endosomes where the pH is very low. So what happens is, since these polymers will have a lot of secondary and tertiary amines, they start sequestering more H plus ions in the endosomes or lysosomes. So there's a lot of H plus ion concentration inside these cells, inside these endosomes and lysosomes. So the polymers will start sequestering these H plus ions. As a result, your pH will start changing inside these endosomes and lysosomes, and as a result, to balance that, more H plus ions are pumped in from the cytoplasm into the lysosomes. Now as more H plus are pumped in, to balance that, more and more chloride ions are also pumped in. So what happens is there's a lot of ionic strength that starts building up inside these lysosomes. So to balance that, a lot of water starts coming in through osmosis, and this leads to bursting of the lysosome. So in this way, your polymers, if they have a lot of secondary and tertiary amines, they help in this is known as a proton sponge effect. They help in bursting open of these lysosomes, and in that way your polymers, the particles then can escape through these endosomes, and your gene can be delivered into the cytoplasm. I hope this concept is clear. This concept of proton sponge effect is clear. So what property of the polymer causes proton sponge effect? It is the presence of your secondary and tertiary amines. Is this clear? Okay, if there are no doubts, I'll move on towards the next question. Next is, which of the following can be considered as an extracellular barrier for gene delivery? We have to choose two correct options. We were just talking about this in the introductory slide, right? That there are a lot of barriers for gene delivery. We could segregate these barriers into either extracellular barriers or intracellular. So here we are supposed to choose two extracellular barriers for gene delivery. So please take your time in going through the options and let me know if anybody knows the answer.
Ma'am, it is 1 and 3.
Okay, 1 is inhibition of transfection by mucus, 3 is degradation of DNA in plasma. Thank you for answering. We'll wait for to see if anybody else has a response, and then we'll discuss the answer. Thanks for answering. I guess nobody else is responding. So let's look at the correct answer. Yeah, you were correct. The correct answer is option 1 and 3. So these are considered as the extracellular barrier. So for more clarification, let's just go through the slide. So as I said, there are various barriers in delivering gene to the cells. Extracellular barriers are considered as degradation of DNA in plasma itself. The moment you inject your DNA solution or your gene solution, whatever it is, if the genes are in the plasma, there are chances of DNA degradation because of the enzymes present in your serum or plasma. The uptake of DNA by reticuloendothelial system, that could happen. Inability to target DNA to specific organs. We want the gene to be delivered to a specific organ. If we are just injecting it without any carrier, more chances that it will get degraded. It will not be targeted towards a specific organ, and it is largely ineffective via the oral route, and the transfection inhibited by mucus. So we have a lot of mucus lining the oral tract, right? The digestive tract. So if we are trying to deliver genes directly to the cells, there are chances that the mucus will inhibit into this process. So your extracellular barriers are inhibition of transfection by mucus and degradation of DNA in plasma. The rest of the options are barriers, like for example uptake of DNA by mitochondria, lysosomal degradation, degradation by intracellular enzymes, or sequestration of gene by histone proteins. These are also barriers, but they are mostly intracellular barriers. So the question is specifically for extracellular barriers. The correct answer is option 1 and 3. Is this clear? Okay, I'll move on towards the next question then. Next is, select the correct options for immune isolated cell therapy. We have to choose two correct options. do let me know if anybody knows the answer.
Is it 1 and 2, ma'am?
One is it can be possible using only xenogenic cells, and the duration of therapy can be prolonged using this method. I think one of your options is correct, option 2. The correct answer is option 2 and I would say 4. So we have to choose what options are correct for immune isolated cell therapy. So what exactly is immune isolated cell therapy? So what we can do is, let's say you have cells which are able to secrete a protein, but you have taken these cells from a different patient's body, from a different human or from a different organism, and you're trying to put it into a patient's body. The patient's body lacks this protein, so you want to deliver these cells which are producing this therapeutic protein. So what will happen is, once you put these cells inside a patient's body, the patient's body is going to consider this as a foreign object, and they're going to launch an immune attack against this. So to prevent that, what we can do is we can use isolated cell therapy. So what we do in this is, we can transfer these cells by encapsulating it into a polymer. So what this polymer layer does, it allows nutrient exchange, it allows oxygen to flow through, because your cells need oxygen and nutrients to stay alive inside this polymer layer, but it prevents the immune cells of the patient's body from recognizing these cells. So in a way, we are isolating these cells from the patient's body, but letting the therapeutic protein go through. So in that way, we can use this concept to treat a disease. So your transplanting cells can secrete a therapeutic protein over a very long period in response to a physiological stimuli. So people have tried using this concept for pancreatic islet transplantation. So these are cells which actively produce insulin. So people have tried using this, and it is used for long term gene therapy using transformed cells. The cells transplanted could be allogenic, or they could be xenogenic. Allogenic meaning from the same species, xenogenic meaning we can take these cells from a different species. Let's say we have taken these cells from a pig's body and then tried putting it into a human body. In that case, that would be xenogenic, and they can so there is chance of generating immune reaction against this. So that's why we put a polymer, we encapsulate these cells into a polymer, which allows nutrient and small molecule exchange, but prevents antibody molecules and T cells from recognizing these cells. So this replaces constant immunosuppression. We don't have to constantly give the patient immunosuppressive agents. That's one advantage. And it also allows xenogenic transplantation. Xenogenic meaning from a different species. So this is about immunoisolated cell therapy. If you just look at the options, first is, it can be possible using only xenogenic cells. That's not correct. We can also use allogenic cells taken from a different human. So that's why this option is not correct. The duration of therapy can be prolonged using this method, yes, as long as your polymer layer is intact, your patient's body is not recognizing these cells as foreign. As long as that happens, the duration of therapy can be prolonged. Polymer encapsulation in which the cells are present does not allow diffusion of nutrients. That's not correct. It does allow diffusion of gases. That's how the cells inside are living. That's how they can survive the immune isolation. So the correct options are, the therapy duration can be prolonged and the polymer encapsulation allows diffusion of gases. So that's why option 2 and 4. Is this clear? Okay. Yeah, I think that's all I have for you for this particular assignment. So we just discussed a bunch of questions regarding gene therapy and so forth. So if you people have any doubts with this particular session, we can discuss it now, or I'll be sharing this video as well as this presentation with you. If there are doubts regarding any other concepts, we can discuss that, or if you want me to go over some of these questions again, I can do that. If not, we can meet next Tuesday at 6 o'clock for the last session. So this was 11th week. We have one more session left in this series, so we can meet next Tuesday at 6 o'clock. If that's okay with you people, I can stop the meeting here.
And can I see the previous questions?
Yeah, sure. I can just show you the presentation again. Nonetheless, I'm also going to share the presentation, but I can do so now. Right, so give me a minute. So you have missed what all questions did you miss? So I'll just go one by one. So the first question was, what is gene therapy? The correct answer is treatment of diseases caused by genetic defect. You can
Okay.
read about this in the slide. Then we started discussing of which of the following cell types are not considered professional antigen presenting cells. The correct answer was neutrophils and T cells, because these are not antigen presenting cells. The antigen presenting cells are macrophages and dendritic cells, and also B cells. So the correct answer was neutrophils and T cells. These are not professional antigen presenting cells. Then we had which of the following are not advantages of non-viral vectors? So the correct answer was high transfection efficiency and natural tropism. These are the advantages of viral vectors. The question is, sorry?
Yeah, these are the advantages of viral vectors.
Right. So the question is, which are not advantages of non-viral vectors? So that's why high transfection efficiency and natural tropism is the answer. You can read about this in this slide as well. Next, which one of the following is not true for chitosan as a gene carrier? You would like to answer this?
It is a cationic biopolymer. It
Right.
is not true, you are asking. Can complex with DNA.
Right.
Um, toxic,
Right. The correct answer is toxic, because chitosan is actually not toxic. That is why it's very,
biocompatible.
yeah, it's very
It is a natural
biocompatible.
biopolymer.
Right. It's a very biocompatible non-immunogenic biopolymer. That's why the correct answer is toxic. Clear? Yeah.
Yes.
Next was, what kind of material is preferred for gene delivery?
Cationic polymers.
Right, correct. Because they are positively charged, your DNA is
Positively
negatively
charged.
charged, so they can form a
DNA is
complex.
negatively charged.
Yes, absolutely right. Were you there for this? What are not the objectives of gene therapy?
The last two.
Yeah, the last two. So we don't use gene therapy to cleave healthy genes.
Yeah.
We don't want dysfunction at a cellular level. Rest of the options, yes, we do use gene therapy for
Correct.
these. Yeah. Then you have, what property of the polymer causes proton sponge effect?
Presence of secondary and tertiary amines.
Right. So that's why we use these polymers, so that they can sequester the H plus ions in the endosomes and lysosomes. In that process, they cause osmotic swelling of these vesicles, which then burst open, and then your polymers, your particles could be released in the cytoplasm. That carries your gene. So that's fine. And then which of the following can be considered as an extracellular barrier? I think you already answered this as option 1 and 3.
Yes.
So yeah,
Yes.
you were there for these questions. Yeah, that's all
Yes.
I have for you. I'll also be sharing this presentation in case you have missed any of the initial explanation, so you can make use of that. That's all.
Last 2 weeks I was not able to attend because it was our Navaratri celebration.
Right. Yeah,
So,
it was a holiday session. So I have already uploaded the PPT and the video as well. You can go and have a look.
in which part, ma'am, your video is there?
This is in the YouTube channel. You people have access to the YouTube channel, right?
Yeah, yes.
Yeah, it's there. Week 9 and 10 is there. 11th I'll upload today. 12th we can have a discussion next week.
Yes, ma'am.
Sure.
But I feel the drug delivery system's full thing, it could have been done into 2 parts. It's very difficult to study.
Right.
Because we are all
So
working people, so we come home only by 5:30.
yeah,
So
that's
we are
true.
finding it very
There's a lot of
difficult
content
to,
in the course. I think we could have
lots
reduced
of
it.
content.
Yeah, the content is a
At
lot,
least it could
but
have been done in 2 parts so that we can have
right.
a nook and corner of everything and learn good.
Yeah.
We
It
are
is a
finding
bit
it very
difficult to
difficult.
grasp all of these concepts, but if you just
Yeah.
keep on reading, I think most of, are you giving the final exam?
Yeah, yes.
Right. So if you just follow all of these assignment questions, I think more or less the final questions would be similar to these. So that
Okay.
shouldn't be difficult for you.
Okay.
Yeah.
Thank you.
Thank you.
Thank you.
Yeah.
Thank
I'll
you.
end the meeting then.
Yeah.
Yeah. Thanks for joining. Bye.
Thank you. Bye.